False negatives can be caused by alignment errors or by obfuscations in the sequence that make it difficult to determine the correct organism or gene. Because many bioinformatics tools rely on heuristics to make alignments faster, tools might miss correct results or alignments or surface incorrect ones. New research can also expose gaps when new genes are discovered or annotated or new variants are found. On the other hand, with sequence obfuscation, a sequence can become so dissimilar from its original that tools might not be able to detect the similarity. Sequence obfuscation is a complicated topic of its own, but some common examples are codon optimization, engineered plasmids, and oligo pool assemblies. These oftentimes have a good scientific purpose, like optimizing yield of a protein or creating a custom construct that might be easier to assemble in-house. We'll go through an example now of an initial DNA screening implementation that misses a controlled region, followed by a reparameterization that corrects this. Here we seem to have a caspase-5 homolog found in a few different primates with relatively high coverage of 72%. From a first glance, this might be a unique protein that probably has some similar functions to the caspase enzyme, but just hasn't been annotated yet. Going to the next example, after reparameterization, we find that there's actually a small 150 base pair region that matches to Rift Valley fever virus with high coverage and high identity. This was initially hidden by the much larger alignments of the caspase enzyme and may have been caused by either too strict thresholds for significance or too much optimization, speed, or power that the heuristics miss the alignment altogether. Because we don't always readily know where a gene starts and ends, we may need to use more conservative measures to account for these kinds of cases. This wraps up the section on DNA screening concepts. It should hopefully give you a good overview of things to watch out for and help guide you through some of the hands-on exercises.